RNA as an essential catalytic role and later as a self-replicating molecule. The basic integrity of a cell is the formation of a cell membrane composed of lipid bilayers. As these can form spontaneously, they could have surrounded early RNA molecules. Their continued presence may have been promoted through mutation of the RNA, which would have been passed on to succeeding generations through self-replication. This basic system does have significant disadvantages because a mistake made in the replication of RNA would immediately have an effect not only on the replication of the genetic material, but also on the ability to act as a catalyst, largely, it may be assumed, in a detrimental manner. The separation of the self-replicating machinery from the enzymes they encode would have resulted in far fewer abortive stages. Consequently, we must assume that DNA largely took over the role of the carrier for the self-replicating genes and proteins of the enzymes that they eventually encoded. RNA merely remained as the messenger that carried the instructions from DNA to the formation of the proteins. The early bacteria emerged approximately 1.5 billion years after the creation of the planet. For the next 3 billion years, bacteria were probably the planet's sole living inhabitants. The fossil record shows that there were huge numbers of bacteria, often collecting in large colonies, attached to many surfaces. Their imprint can still be seen. Approximately 1 billion years ago, however, the numbers of these colonies in the fossil record began to fall, and this could be evidence that bacteria had become the source of food for some other life form. Certainly this was before the Cambrian explosion of 500 million years ago, when the diversity of life forms increased rapidly, but it does suggest that for the first 3/4 of life on Earth, bacteria had it all their own way. What were these bacteria? The common view is that prokaryotic cells, such as bacteria, and eukaryotic cells, such as those that comprise our bodies, had a common ancestor. The last universal common ancestor, Luca, or senancestor, is considered to be the most recent ancestor of all life on Earth, and possibly was living some 3.5 billion years ago. It is thought to have been a prokaryotic, single-celled organism, possibly similar to simple bacteria found today. By this time, it has been concluded that the genetic code must have become DNA rather than RNA, and that the catalysts had become true enzymes, proteins, composed of the 20 amino acids. Furthermore, the machinery for dividing the DNA, maintaining its integrity, and expressing the genes through RNA was already established. These bacteria would have been exclusively anaerobic. They did not respire oxygen, as there was little or no available oxygen in the atmosphere at the time. These bacteria could produce energy from the available nutrients, but this was an extremely inefficient process. About 3.2 billion years ago, photosynthetic bacteria, or cyanobacteria, first emerged. These bacteria could use energy from the sun to make sugars, which were used for further metabolism. The byproduct of this photosynthesis was oxygen, which began to accumulate in the atmosphere. Oxygen is toxic to many cells, particularly the early anaerobic bacteria, which probably started to decline. About 2.5 billion years ago, the fossil record shows that aerobic bacteria emerged, able to use the newly available oxygen to convert sugars into energy, usually in the form of adenosine 5-triphosphate, ATP. The use of oxygen vastly increased the energy obtained from a single sugar molecule, and these bacteria soon predominated. The main eukaryotic cells may have derived from an early example of archaea, which themselves derived from Luca. They probably evolved about 2 billion years ago. These were distinguishable from prokaryotic cells by having a defined nucleus, usually comprising discrete chromosomes that contained the DNA. However, having emerged from prokaryotes, the evolution of these eukaryotic cells did not proceed independently of prokaryotes, but with a degree of symbiosis. Eukaryotic cells possess distinct organelles. Most animal cells possess mitochondria, and most plant cells contain chloroplasts.